Hello and welcome to my channel Sam Biology Class. Friends, today we will discuss about the principal advantages and disadvantages of bright field microscopy. But before that, will you please consider subscribing my YouTube channel? Your subscription and viewership are often a lot to me. Bright field microscopy is the most common light microscopy technique where the specimen appears dark against a bright illuminated background. This technique utilizes transmitted white light from a specific source such as a halogen lamp. Most of the biological samples are colorless because their constituents, primarily water, protein and lipid, do not absorb visible light. Instead, they allow all wavelengths of visible light to pass through directly without being significantly absorbed or deflected. As a result, the specimen appear nearly invisible or have extremely low contrast. Because of this, the sample blends into the background. As a result, it becomes difficult to visualize structural details and the specimen appears white or transparent. We know that most of the cells are composed of 75% of water, which is colorless. Unlike plant cells which have chloroplasts, many animal cells and microorganisms do not contain pigments that can absorb light in the visible spectrum. So when such type of transparent samples are focused with white light in bright field microscope, most of the light passes through the sample directly without being significantly absorbed or scattered. This gives rise to very low contrast. As a result, fine structural details are lost and the specimen and the background appear almost the same. Now we came to know about the reasons why transparent samples are hard to see with bright field microscope directly without pre-treatment. Now the question is how to visualize them. They can be visualized by fixing them on the slide and treating them with specific color reagent known as stain. Different stains impart different colors used for different types of samples for different purposes. For example, gram stain is used to differentiate between gram-positive and gram-negative bacteria. Giemsa stain is used to color blood cells and to identify parasites such as Plasmodium. Hematoxylin is used to color cytoplasm, and eosin is meant for highlighting cytoplasm and extracytoplasmic matrix. In order to visualize, the sample is first fixed on a slide and then treated with a suitable stain for a specific time period. After incubation, the slide is washed in order to remove excess stain, and then the slide containing sample is placed on the stage. And this is the sample. Key components of a bright field microscope are illuminator. It is the source of light, usually a halogen lamp. It transmits white light. Second component is condenser. Another component is stage on which the slide is placed. Next component is an objective lens. And final component is the ocular lens or eyepiece. So when the stained specimen is placed on the slide and the illuminator is switched on, it transmits white light. The transmitted white light from the source is then collected by the condenser, which then forms a solid cone of light that is focused onto the specimen. The transmitted light coming from the illuminator is collected by the condenser lens, which focuses a solid cone of light onto the specimen. In addition, some of the light passes through the surrounding. As a result, the background appears bright. When the light passes through the specimen, it is absorbed, refracted or scattered up to different extents by different regions of the specimen having different densities. This differential absorption, refraction and scattering of light by specimen increases the contrast. As a result, the image becomes clearer, sharper and more detailed. Then the light passing through the specimen and passing directly through the surrounding is collected by the objective lens, which forms the real magnified inverted image, which is used as an object by the ocular lens. The image formed by the objective lens is further magnified by the eyepiece that produces final virtual upright and further magnified image. Bright field microscopes are widely used in histology, cytology and microbiology for visualizing stained tissue sections, cells and bacteria. Advantages: bright field microscopes are easy to operate with minimal training, making them ideal for education and routine lab work. It is significantly cheaper than fluorescence or confocal microscopy. This technique allows visualization of natural pigmentation in sample and easily adapted with cameras for digital imaging and image analysis. Disadvantages: unstained, transparent, or living biological samples are difficult to see with bright field microscope due to low contrast. Therefore, it requires staining or fixing samples for high contrast, which can kill cells and introduce artifacts. In addition, resolution of bright field microscope is lower than electron microscope, thereby limiting visualization of very small structures like viruses. So friends, this is all about the principle of bright field microscope, advantages and disadvantages. For more updates, subscribe my YouTube channel, Sam Biology Class.